right, let's talk about the humoral response. All right, we're talking about B cells, and we're talking about extracellular pathogens. Okay, so keep our colors. All right, so we have our B cell, and this is a naive B cell, and it's going to be going over to, like some, lymph node or something. It's going to start looking for its antigen, and the antigen, it's this extracellular pathogen, so these are going to be, antigens that are going to, like bacteria that are floating around in the fluids in the blood. Okay, because remember, this naive B cell is in the lymph node, and what's coming through there is lymph. Where does lymph come from? Blood. Okay, so the pathogen then comes around and it's going to bind. Remember, it's the exact shape. Okay, and so it's going to bind to the antibody. So this thing is like a marker. So let's say this is the bacteria right here, and this is just one of many markers that are hanging off of here. So this is the antigen. This is the one that this naive B cell sees. So it binds to that. So is that enough? Well, no. We know that this cell has to be activated. So when this cell, naive B cell, binds to that antigen, basically what it does is it endocytoses this whole thing. This whole complex gets endocytosed into the cell. And basically, it kind of breaks down that thing and the pathogen thing into its sort of protein parts. And what it does then is it takes those protein parts and posts them on a platform. And that platform, of course, is going to be an MHC2. Because remember, B cells are antigen presenters. So they have MHC2 platforms, not one. Okay, so now it's there and it's posted on its little platform. Is it activated? No. To activate, it must have the help of a T helper cell. Okay, so this is a T helper cell that has this little T cell receptor, and it's going to bind with the MHC2 platform of the naive B cell. So it's going to bind there. It's going to link up. Is that enough to activate this cell? Well, guess what? It really isn't quite yet. So the T helper cell then sends over some chemicals. And these chemicals they're cytokines. And they're cytokines called interleukins. And that officially activates the cell. Okay, so now we have activation. Okay? So what happens in activation? Well, what happens in activation is something called clonal expansion. And this is where the B cell divides. And it starts dividing and it produces plasma cells. So plasma cells are these cells kind of like that, sort of, and they are antibody makers. So they make antibodies. And they make antibodies with that receptor site. Okay? And they make a ton of them. They barf out about 2,000 antibodies per second. So on and on and on, and about 2,000 per second there. All right? They also make memory cells. That is not how you spell memory. I am not a speller. So these are memory cells. So these cells remember what happened. They will come into play if we get exposed to this antigen again. Okay? So the original response is slow. It takes a while to do all this stuff. It takes a while to become activated and find the T cell with the same. It just takes a while. Okay, so it takes a bit, and it's a very strong response. Okay, this response, the memory response. So the second or third exposure, it's fast because these guys already know what to do. They don't have to go through all this activation stuff. It's fast and it's even stronger, the response. All right, so next we'll talk about T cells and cell-mediated immunity.